Hello, everyone. Welcome to today's live broadcast, Liquid Biopsy and NGS, Moving from Sample to Insight. I am Julianne Chiat of LabRaed, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRaed and sponsored by QIAGEN. QIAGEN offers you the most reliable technology for sample isolation because samples matter to your success. Our top quality assays and panels enable you to accurately analyze and identify diseases and genetic variations. Our informatics software and curated knowledge bases transform your raw data into relevant, actionable findings. QIAGEN is the only company today that offers the complete sample-to-insight solution. Partnering with QIAGEN can make your work a difference. For more information, please visit www.QIAGEN.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You could submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right hand of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. I would now like to introduce today's featured speaker, Raed Samara. Dr. Raed Samara is a global project manager for NGS technologies at QIAGEN with a focus on pre-analytics and targeted enrichment. Prior to joining QIAGEN, he was postdoctoral fellow at the National Cancer Institute, conducting research in the field of cancer immunology with an emphasis on identifying strategies to boost the efficacy of cancer vaccines. He received his PhD from Georgetown University in tumor biology. I will now turn it over to Dr. Samara for his presentation.
Thank you, Julian, and thank you everyone for joining this webinar. What I would like to go through today as just a list of topics. First, I want to introduce the idea of liquid biopsy for precision medicine. Then get into the challenges associated with liquid biopsies and how we have put together solutions to address these challenges. Then the third topic is to give you examples of how liquid biopsies are used. I will go over an example from a recent paper that have used liquid biopsies to monitor disease progression and identify mutations associated with resistance to therapies, and then close this webinar with summary and questions. So on to the first topic, liquid biopsy for precision medicine. So what is the idea of precision medicine? It is the right drug for the right patient at the right time and at the right dose. On the left side of this slide, you see this one-size-fits-all approach. We know that this approach does not work for everyone. And on the right-hand side, you see the precision medicine approach. So on the left-hand side, the one-size-fits-all approach, this is the approach whereby patients are given, the same patient population is given a specific therapy. This translates into three different consequences, basically a positive effect where you see actually the patient responding to that specific therapy, then you have no effect, or you can end up with adverse effects. The reason why the one-size-fits-all does not work is because of all these consequences. What we would like to achieve is for all these patients to have this positive effect and avoid the no effect or adverse effect consequences. The precision medicine approach uses genetic profiling, mainly from liquid biopsies. You could also use it on solid biopsies, but for the sake of this webinar, I'm going to focus mainly on liquid biopsies. So the idea is to extract genetic information from liquid biopsies to then guide us to a specific therapy that will have a positive effect for this patient population. So now, what types of variants can we use and can we detect for precision medicine? Just as three examples, I'm showing you here the mutation type of DNA variant, insertions and deletions, and copy number variations. Now, the knowledge into these types of variants is extremely important, not just to only understand what is happening at the molecular level of that disease, but also we can use this information now to enable precision medicine. Now, how can we do this? Basically, if we look at an example for mutations, for instance, we know that the BRAF V600E mutation is present in a lot of melanoma cases, and this mutation drives the development and progression of melanomas. But the good news is that we can use now that information for a specific therapy. There has been a therapy called vemurafenib, or PLX 4032, that has been developed to specifically target and address the mutation, the BRAF V600E mutation in melanomas. As an example for insertions and deletions, we know that there are a lot of deletions in the EGFR gene that are present in lung adenocarcinomas. The good news is that we can target these types of variants with specific tyrosine kinase inhibitors like erlotinib and gefitinib that will actually target these types of variants and result in good responses. For copy number variants, an example that exists is the HER2 amplification in breast cancer. Again, the good news is that you can actually target these copy number variants with trastuzumab with very good efficacies. So the reason I bring this up again is just to kind of show you the importance of understanding the types of variants that exist in biological and clinical samples for precision medicine. Now, how many variants do you need to look at, and what kinds of variants exist out there that will enable us to take this precision medicine approach? Anyone who has done NGS understands that you can end up with millions of variants or millions of mutations in the DNA. Not all of these mutations are actionable. Not all of them can actually give us information about that specific disease and information that we can use for the management of that disease. You can end up with millions of common variants. These are variants that are present in me and anyone else who's on this call. A lot of these variants do not actually do anything, so these variants need to be filtered out of that process. There are a lot of variants that have no obvious effect on a protein, and so these variants also need to be filtered out. So you start off with millions of variants, and then once you go through this filtering process of removing common variants, removing any variants that have no effect on the protein, and go through this iterative biological interpretation approach, what you end up with is probably tens to hundreds of biologically relevant actionable variants. Again, these are the variants that I mentioned previously, like the BRAF V600E mutation. This is an actionable mutation that you can use for disease management and understand what is happening at the molecular level of that disease. So after going through this process, you end up eliminating a lot of variants that basically have no actionability associated with them. And to give you an example from the literature, this is data taken from a paper that was published back in 2012 in Cell. It might look outdated, but it actually drives the point that I'm trying to make here. The authors here looked at the current lung cancer biomarker landscape. They wanted to understand what types of variants and mutations are accumulating in lung cancer specimens. And what they found is that 25% of the mutations accumulating in these specimens fall within the KRAS gene, and 23% fall within the EGFR gene. And then you have these smaller types of mutations accumulating in a smaller number of specimens. Why is this information necessary and important? Again, because now we know what is happening at the molecular level. We know that KRAS and EGFR mutations actually drive the development and progression of lung cancer. But it is also extremely important from a disease management perspective, because we know that individuals who harbor this specific EGFR mutation, the L858R mutation, they show very good responses to tyrosine kinase inhibitors, again, like the tyrosine kinase inhibitors that I mentioned earlier. But at the same time, if the same individual harbors mutations in the KRAS gene in the form of G12C, we know now that the responses drop dramatically. So while you have a good response for patients or individuals who have this L858R mutation, the responses now drop dramatically if you see an additional mutation in the form of KRAS G12C, just as an example. So again, it is extremely important to understand what is happening at the molecular level, but also you can use that information for disease management. Now, the question is that always comes up is, how do you test for these mutations? Do you take a sequential testing approach where you test for each one of these mutations individually? This is what's been done so far before the introduction of NGS into the clinical research area, where individual qPCR assays were used to interrogate all the different mutations in the KRAS gene or EGFR gene or and so on and so forth. The disadvantage of this approach is that it takes a long time to rule out these mutations, and it also takes up a lot of the DNA sample. And if you have a sort of a liquid biopsy sample, you might not be able to actually look at all these mutations in order to determine what is going on with that sample. The alternative approach is parallel testing, where you test for all of these mutations in the same assay in the same run. And that's where NGS comes in specifically for liquid biopsy, because you don't have a whole lot of DNA. You want to use one assay and you want to run it in the same sequencing run so that you're preserving a lot of that sample. You could use NGS very efficiently to look at all the mutations associated with these genes in one sequencing run using one DNA sample. The idea of liquid biopsy is not new. We've known for a while that we can actually use DNA, cell-free DNA present in plasma to look at mutations. This is a paper that was published back in 2012 that nicely shows how liquid biopsies can be used to screen for and identify low-frequency mutations, to non-invasively identify the origin of a metastatic relapse, and to monitor tumor dynamics over time. So the idea is not new. It's just what is happening now. There has been a lot of developments within the liquid biopsy field that allow us to accurately call specific mutations within cell-free DNA for these applications and purposes. And again, towards the end, I will show you an example from the literature of how a group at a pharma company used liquid biopsy approaches to identify mutations associated with resistance to tyrosine kinase inhibitors. So, but how can you look at DNA variants in liquid biopsy samples? There are three main levels for NGS. You have the whole genome sequencing approach, you have the whole exome sequencing approach, and the targeted DNA sequencing approach. There's no one right or wrong answer as to which approach should be taken. I guess the answer depends on what you're trying to accomplish with sequencing. The main differences when we look at these three different approaches, they differ on several parameters or attributes, and what I try to do here is to compare them on several attributes. If we look at the information level, basically what can you use this sequencing approach for? With a whole genome sequencing approach, you basically look at everything. You're looking at the 3 billion bases in one's genome. With a whole exome sequencing approach, you're only looking at the bases that code for proteins, right? And with a targeted DNA sequencing approach, you as a researcher, you're defining the regions that you want to look at. Why is this important? Simply because if you're looking at everything with a whole genome sequencing approach, this might be extremely helpful if you're trying to discover new variants. But remember that a whole genome sequencing approach is still very expensive. So on a cost per sample, at a minimum, you're paying $5,000 or so to do a whole genome sequencing approach. With a whole exome sequencing approach, this drops down to maybe $2,000. But with a targeted DNA sequencing approach, because as you will see in a bit, you can multiplex many samples simultaneously in the same sequencing run, you're able to decrease the cost per sample. Now, detection of low-frequency mutations, this is extremely important if you're looking at cancer samples, because we know due to tumor heterogeneity that some mutations will be present at very low frequencies within that sample, and you need an approach that allows you to detect these low-frequency mutations. With a whole genome sequencing approach, because you're not able to achieve a very high degree of coverage across each one of the bases that are covered, you might not be able to detect these low-frequency mutations. With a whole exome sequencing approach, because you've increased the depth of coverage associated with these types of sequencing runs, you might be able to detect these low-frequency mutations. But with a targeted DNA sequencing approach, because you can achieve a very high degree of depth of coverage, you're able to detect these low-frequency mutations. They also differ in terms of DNA inputs. So for a whole genome sequencing approach, you require at least 1 microgram of DNA in order to accurately run a whole genome sequence. This might be prohibitive for certain samples, like FFPE samples, and definitely for some liquid biopsy samples, and you might not have enough to take this whole genome sequencing approach. Whole exome sequencing requires less DNA, but it's still prohibitive for some liquid biopsy samples. For the targeted DNA sequencing approach, you can drop this requirement down to 10 nanograms or so of DNA, which now makes it very feasible to look at liquid biopsies using NGS. The number of samples multiplexed is also going to differ because you always have a limited capacity with NGS. So while it is a very powerful tool, you're still limited by the size of the flow cell or the chip that you're using. So when you look at a whole genome sequencing approach, you might be able to run, at least one sample per sequencing run, but definitely not more. So you're taking all those costs associated with the sequencing run and just splitting them on one sample. With a whole exome sequencing approach, you might be able to fit 2 samples. But with a targeted DNA sequencing approach, you can fit up to 96 or even 384 samples or so, which decreases the price per sample. So when you look at the benefits of targeted DNA sequencing, it gives you more relevant data because you as a researcher, you're defining the regions that you want to look at. It is more cost effective because you can multiplex many samples simultaneously. The cost per sample decreases. It gives you the ability to detect these low-frequency mutations, and it requires a lot less DNA than other approaches. So when we think of liquid biopsies, targeted DNA sequencing approaches are much more attractive than whole exome or whole genome sequencing approaches if you're trying to decrease the cost, if you want to achieve more coverage, and be able to detect these low-frequency mutations. So why choose a targeted DNA sequencing approach? As I mentioned earlier, it gives you a very well-defined content, so you can examine those variants that matter to the disease that you're studying. It offers you a small target size. You, as a researcher, you're defining this target size, which allows you to multiplex many samples simultaneously to save money. And it gives you more reads per sample, so you can achieve a higher degree of depth of coverage to allow you to detect these low-frequency variants. So just to kind of put this into a visual, with targeted DNA sequencing approach, what you're trying to do is to shrink the genome. So instead of looking at every base in that genomic DNA that you're studying, you're shrinking that DNA down to maybe 3 or 4 or 5 or 50 or 100 genes that are relevant to the disease that you are looking at. Just as an example, I'm showing you here 3 genes, KRAS, EGFR, and IDH1. With a targeted DNA sequencing approach, what you're going to do is you're going to enrich these targets. So you use different technologies. I'll show you what we have put together for targeted enrichment, which is multiplex PCR. But you could use multiplex PCR to enrich these genes from the genomic DNA that you're studying. And what you end up with is instead of sequencing every base, you're going to sequence only the bases contained within those regions that you've enriched. So if I enriched KRAS, EGFR, and IDH1, the variants that I'm going to be looking at are those variants contained within KRAS, EGFR, and IDH1. Again, this is a very attractive approach if you want to decrease your costs and be able to detect these low-frequency variants. However, the idea of liquid biopsy and using targeted DNA sequencing is very attractive, but there's still a lot of challenges associated with liquid biopsy approaches. And what I will do next is to go through 3 main challenges that we see for liquid biopsies. And the 3 main challenges, first is low DNA quality. We know that the DNA obtained from liquid biopsies is highly fragmented. You end up with very small fragments, and you need an approach that allows you to actually use these small fragments instead of this highly intact DNA that you isolate from your samples, like FFPE samples or frozen samples or so. The second challenge is the low DNA quantity. You're not going to end up with micrograms of DNA. You're going to end up with low nanogram ranges, and again, you need a technology that allows you to use that amount of DNA, very low quantities of DNA for enrichment and library construction. Another challenge is the presence of low variant allele frequencies. So again, this goes back to the idea of tumor heterogeneity. If we want to focus on cancer, we know that some of these mutations are present at low frequencies, maybe down to 1 to 5%, and you need an approach that allows you to detect these low frequency variants. So what I will go into next is to address each one of these challenges that show you how our solutions can address these challenges. So on to the first one, low DNA quality. As I mentioned earlier, there are multiple ways by which you can do enrichment or target enrichment and library construction. Mainly, it is hybridization-based approaches or multiplex PCR-based approaches. We chose to go with multiplex PCR-based approaches because it basically delivers unmatched specificity and uniformity. This was nicely shown in a paper published back in 2010 in Nature Methods that compared multiple target enrichment strategies or approaches, and their conclusion is that the specificity of PCR will almost certainly always exceed that of hybrid capture, and its uniformity may never be matched by either hybrid capture or MIPs. So we chose multiplex PCR, again, because of the high degree of specificity and uniformity that multiplex PCR strategies offer. So what is multiplex PCR all about? The idea is very simple, actually. It all starts with defining your genes. And just as an example, I'm showing you here gene 1 and gene 2 just as an example. So let's say I am interested in gene 1 and gene 2 in that disease that I'm studying. Call this BRCA1 or BRCA2 or so. The next step for multiplex PCR is to design primers that will generate amplicons that correspond to gene 1 or gene 2 or BRCA1 or BRCA2. These primers are designed in a way to generate either smaller amplicons or maybe a little longer amplicons. But again, the idea is to use all of these primers to now enrich gene 1 and gene 2. Once we've designed primers against these targeted regions, then we use our primer separation algorithms to separate these primers into an appropriate number of PCR pools. The number of PCR pools can range between 1 and 4. And what determines the number of pools is just how far apart these primers are. We want to keep them separated in a way to produce a uniform range of amplicons. We generally target 150 base pair amplicons because we want them to be small so that they're compatible with cell-free DNA. And so we will, after designing these primers, we will separate them into an appropriate number of PCR pools. So this is what the chemistry looks like. You have your primers separated into an appropriate number of PCR pools, and then you have the PCR chemistry that now allows you to take these primers, combine them with the genomic DNA that you've isolated, run a simple PCR setup, and now you've enriched for those two genes. Again, just as an example, you've enriched for those two genes that you're interested in. I mentioned that the amplicon size that we generate is 150 base pairs, so it is small enough to work with the highly fragmented nature of DNA from cell-free biopsies. But at the same time, it gives us enough information to accurately map and align those reads coming from the sequencer back to that reference genome that we'll be using. So it is small enough to work on highly fragmented DNA, and it gives us enough information to basically analyze the data. So when we look at the low DNA quantities, I mentioned that another challenge is the amount of DNA that you isolate from cell-free DNA samples. And in order to tackle this problem, the way we developed our panels, the GeneRead DNA-seq panels, is to allow them to only require 10 nanograms per PCR pool. So if you think of a panel that has 3 PCR pools, the requirement for DNA is only 30 nanograms total. So you have 10 nanograms per PCR pool, which now makes this technology and these products compatible with the low amounts of DNA that you get from cell-free DNA. Looking at low frequency variants. So again, you want to be able, especially if you're looking at tumor tissues, you want to be able to detect those low frequency variants. So what in order to tackle this problem, we've taken a sort of an application-specific amplicon design. Remember that for target enrichment using multiplex PCR, you're going to design these primers that correspond to the specific regions that you want to enrich. So in order to ensure that these primers will work efficiently and to avoid as much as possible any dropouts associated with multiplex PCR, what we've taken is this like redundant tiling approach or redundant overlapping approach for amplicon design. Just as an example, I'm showing you here 2 different designs that we have taken where to highlight this overlapping approach. What you see in blue here, you're looking at here just an IGV snapshot of the KRAS gene as it is covered by the actionable mutations panel and the clinically relevant panel that we've developed. And in blue and red, you're looking at the amplicons that will be generated from the primers that we've designed against this specific region of the KRAS gene. So I'm specifically looking at the G12 locus of the KRAS gene because it is an actionable variant, as I mentioned previously. So for the actionable mutations panel, the way we've designed this panel, because it is a hotspot panel, and we want to ensure that every hotspot gets covered regardless of the sample type that you're using, especially for cell-free DNA or liquid biopsies, we've designed 4 different amplicons to cover this specific locus within the KRAS gene. By doing so, even if one of the amplicons drops out for whatever reason, we still have 3 other amplicons covering the KRAS G12 locus to accurately and confidently call any variants that fall within this region. Another approach that we have taken is to avoid any edge effects or blind spots. What do I mean by edge effects or blind spots? When it comes to multiplex PCR sequencing, one of the main issues with multiplex PCR sequencing is that the ends of the amplicons do not get sequenced very efficiently. So if we look at the 1 to 2 bases that exist at the ends of an amplicon, we know that these bases do not get sequenced very efficiently. It's something that we call edge effects or blind effects. In order to circumvent this issue or address this issue, what we do is instead of removing the primer sequences before the sequencing run, we actually keep the primer sequences during the enrichment and library construction and sequencing, and we only remove the primer sequences before the variant calling step. So we're not calling any variants within the primer sequences, but we keep the primer sequences to protect the ends of the amplicons so we don't miss any base within those amplicons. Because remember, the amplicons represent those regions that you're interested in, and you do not want to miss any of the bases within that region that you're interested in. This was nicely shown by a group in Germany that compared 2 different approaches for multiplex PCR, the QIAGEN approach and the AmpliSeq approach. So they did the enrichment and library construction using the 2 different workflows, and then they sequenced the libraries on a MiSeq. Now, when they compared the sensitivity and specificities of the panels and the workflows, what they show in their paper is that 77 out of 77 mutations were called using the QIAGEN panel and workflow. However, only 69 out of 74 mutations were called using the AmpliSeq workflow. So they ended up missing 5 mutations using the AmpliSeq panel and workflow compared to the QIAGEN panel and workflow. The reason is because these mutations happen to be at the amplicon boundaries and primer binding sites. Again, because the AmpliSeq workflow removes the primers physically from the amplicons before the sequencing run, you end up exposing the ends of the amplicons. And if the mutation happened to be at the ends of the amplicons, you're not going to sequence it efficiently and you won't be able to see it or analyze it. So keeping the primer sequence is extremely important. And now if you're looking at these low-frequency mutations, you want to capture every single base that you can. And in order to do so, you want to protect the ends of the amplicons and keep the primer sequences and only remove them right before the variant calling step. Another challenge is if you have two different sequencing platforms. We know that the use of 2 sequencing platforms can be useful if you want to validate the results that you're seeing on one sequencing platform on another sequencing platform. So we've built the panels and to be a platform-independent solution. So you can use the same box, the same panel, whether you're looking at the Illumina platforms like a HiSeq, NextSeq, or MiSeq, and you can use the same panel if you're using the Ion Torrent platforms like a PGM or Proton. So it's a platform-independent approach to allow you to use the same content, the same primer sets, regardless of the sequencing platform that you're using. So we put together several different panels, as you can see on this slide here, for different types of applications, for solid tumors, hematologic malignancies, tissue-specific content, gene-specific content, as well as comprehensive content. You see the list over here. All of this is also available. All of this information is available at QIAGEN.com. If you want to learn more, please visit QIAGEN.com, and all you have to do is just type in DNA-seq panels, and it's the easiest way to find these types of products. Now, where did this content come from? The content came from many different databases, such as the Cancer Genome Atlas, NCCN, Cosmic databases, specific reviews within Nature and in Science, and we even tapped into private databases like the Ingenuity database from QIAGEN to build this content and put it together. If you don't like the content for any reason, you can build your own panels using two different offerings, the mix and match and custom offerings. The mix and match gives you access to 570 bench-tested gene designs. The custom panel gives you access to pretty much the entire genome. You can define your content in terms of genes. You can define them in terms of genomic locations. We will use our primer design algorithms and primer separation algorithms to put together any content that you're interested in. All you have to do is just put a list of your targets together and use the custom panel builder that's present online to build your custom panels or the mix and match panels. But to show you the power of mix and match, so I mentioned mix and matches are these 570 bench-tested gene designs. They were tested at the bench and they were optimized to give you a high degree of experimental coverage and uniformity. And anyone who has used any multiplex PCR-based approach understands that if you add one primer to that mix, you can alter the performance of those gene designs. So what we wanted to do with the mix and match is to optimize the gene designs as much as possible so that the same gene design will give you consistent performance across different panels. So regardless of the panel that gene design exists in, you'll see the same type of performance. So just as an example, if we look at the MET gene design, the MET gene design exists in 4 different panels, as you see on the x-axis. So all these numbers 004, 008, they represent different panels. But if we look at the MET gene design in terms of experimental coverage and uniformity, we do see a very high degree of sort of consistent performance of the MET gene design regardless of the panel in which it sits. So whether it sits in the comprehensive cancer panel, which is the largest panel that we have, the 501X panel, or one of those tissue-specific panels like the 004, which I believe is the lung cancer panel, you do see a consistent type of performance for the same gene design regardless of the panel in which it sits. So just to kind of go over the challenges or the features and the benefits of the multiplex PCR-based approach that we've put together for target enrichment, specifically the GeneRead DNA-seq panels. Again, it offers you small target sizes. You can go as small as you want, and you can go up to maybe 200 genes or so. And the benefit is that it gives you can multiplex many samples simultaneously to save money. It uses multiplex PCR, so you can achieve this very high degree of specificity and uniformity compared to other approaches. It offers you a high degree of coverage depth or depth of coverage, so you can be more confident in calling those low-frequency mutations. Low DNA input material. This is specifically, again, for liquid biopsy approaches, so that you can sequence those low-yielding samples, such as liquid biopsy samples. Quick turnaround time, so you can go from sample to insight quickly. It takes about 8 hours to go through the enrichment and the library construction step, so you can turn a genomic DNA into a library that is ready to be sequenced in about 8 hours or so. No physical removal of primer sequences, as I described earlier. That way you can reduce the false positives and negatives and achieve that 100% specificity and sensitivity. Platform-independent solution, so you can use the same panel for different sequencers. We pool the amplicons before the library construction step, which saves you money, so you don't have to prepare libraries from individual PCR pools. Instead, you are preparing libraries, one library per sample. Because we pool the amplicons after the target enrichment step and before the library construction, we end up with only one library per sample. And it is an integrated workflow in the sense that the bioinformatics tools have been coupled with the regions that get enriched, and so you can sequence those low-quality samples. So looking at data analysis, I mentioned that it is an integrated workflow whereby the data analysis tools have been optimized to also look at amplicon-based sequencing. And just to kind of give you a sense of the tools that have been developed for data analysis, we know that there are several layers of data analysis from primary, secondary to tertiary. What we've put together for the secondary level of analysis is the variant calling platforms, and then for tertiary analysis, it is more on the interpretation side. So once you determine what types of variants are present in your sample, how can you interpret that information? What does it mean basically to see a specific mutation in that sample? And to kind of just give you a sense of the different tools for bioinformatics, we have the CLC platforms that allow you to, again, take the FASTQ files or BAMP files and determine the different types of variants that have been detected in those samples. It is an easy to use tool. It doesn't require a whole lot of knowledge of bioinformatics. You need some background in bioinformatics in order to construct these pipelines. But even if you don't, we have a team of scientists that can help you build this pipeline. But what is also great about this tool is that there is a plug-in for the GeneRead DNA-seq targeted panels. Once you double-click on that plug-in, it basically builds this pipeline to allow you to analyze data coming out of the GeneRead DNA-seq panels. So when I mentioned that it is an integrated workflow, it actually has built specific tools to allow you to do amplicon sequencing coming out of the GeneRead DNA-seq targeted panels. So you don't have to build this pipeline anymore. It is already pre-built for you. Another feature is that it is a flexible tool in the sense that you can change the parameters, you can change thresholds to fit the type of application that you're using NGS for, but at the same time, you can also lock the design or lock the pipeline so that no one can change it. This could be extremely important if you have a specific workflow and that has to be applied across all the samples that you're using. You want to make sure that you lock that design or pipeline so that it doesn't get changed, and you can compare the data coming out of the samples very easily. So once you've identified the variants, what can you do for interpretation? There are multiple tools. The one that I will highlight over here is the Ingenuity variant analysis. It allows you to rapidly prioritize and annotate these variants, and also you'll be able to actually see the supporting publications that support this specific variant and why it is important for that specific panel, sample that you're using. So you see on the left is this filter cascade, whereby you are filtering the variants from basically starting off with all the variants that have been detected down to what is predicted to be deleterious variants. So remember, earlier in this webinar, I went through sort of this filtering cascade whereby you're starting off with millions maybe of common variants, depending on the regions that you're looking at within the genome. But then you have to, in order to determine which ones are important, you have to go through this filtration cascade. And that is what you see on the left-hand side. So you're starting off with a large number of variants, and you end up with maybe only two variants that are predicted to be deleterious. And the two variants that you see happen to be BRCA1 and BRCA2 variants, as you see in the middle of the slide. So the Ingenuity variant analysis tool is going to allow you to go through this filtration process, and it's going to show you those variants that are extremely important or that are predicted to be deleterious. And if you want to then further understand the importance of these variants for that specific disease that you're studying, it links all this information to all the papers that have been used to annotate that specific variant. And you can also get a nice visual of where these variants sit within that gene that you're looking at. So it's a way to interpret the variants coming off of the CLC platforms. So when we look at how targeted DNA sequencing is used, we mainly see it in the clinical research area where you're interested in detecting specific variants that are known to exist, but also have sort of a limited capacity for discovery. Just because you're using panels doesn't mean that you cannot discover variants. As a matter of fact, there have been quite a number of papers that have used specific panels, whether they are small or large, to identify novel variants. So you can use a panel to identify novel variants, but the main application for panels is basically to detect variants that are known to exist. So the third topic that I want to touch upon is to give you an example of how customers have used liquid biopsy approaches for the identification of variants associated with resistance to specific therapies. So I want to go back to lung cancer because this is it's probably the best disease to study when it comes to variants. We know that there are a large number of genes associated with the development and progression of lung cancer, and there are a lot of variants within these genes that are extremely important to identify. So when we look at the landscape of resistance mutations in non-small cell lung cancer, so let's say if we look at how mutations develop and how we can use those mutations for the development of specific therapies, we know that EGFR mutant lung cancer is a subtype of non-small cell lung cancer that exhibits sensitivity to EGFR tyrosine kinase inhibitors. So certain mutations exist within these samples. We know we can target them with tyrosine kinase inhibitors such as erlotinib and gefitinib. However, we know that acquired resistance is going to develop after a median of 9 to 14 months. So you have these individuals on tyrosine kinase inhibitors. After 9 to 14 months, you'll start seeing resistance develop against these, call them first-class tyrosine kinase inhibitors. The most common mechanism of tyrosine kinase inhibitor resistance is a second site mutation within the EGFR gene called the T790M mutation. So after 9 to 14 months of being on these tyrosine kinase inhibitors, what we generally see is that these individuals will start developing this T790M mutation. And that accounts for the majority of resistance against the first-class tyrosine kinase inhibitors. So the idea was, why not develop a specific therapy or drug that targets the T790M mutation? And let's call it the second-class tyrosine kinase inhibitors. This is an approach that a lot of people have taken, and it actually works. So one of the examples is AZD9291, which is an oral, irreversible, mutant-selective EGFR tyrosine kinase inhibitor that has been developed to specifically address those mutations, the T790M mutation within individuals who have been treated with the first-class tyrosine kinase inhibitors. So again, you see that this mutation starts accumulating in individuals. Well, why not develop a specific drug that specifically targets the T790M mutation? That has been successful. Now, with the paper that I want to go into, the authors basically pose this question: can we identify potential mechanisms of resistance to AZD9291, which is the sort of the second-class tyrosine kinase inhibitor that has been developed against T790M mutations? Can we identify potential mechanisms of resistance for this drug before the availability of a resistance biopsy specimen? Goes back to the idea of liquid biopsies. Instead of waiting for a solid sort of resistance tumor to come back, can we use blood samples to identify any resistance mechanisms that could develop after administering this type of drug to individuals? And that is what is described in this paper that was published in Nature Medicine back in May of 2015. So the authors wanted to, again, just ask the question, can we identify any potential mechanisms of resistance using liquid biopsies? So what they did is they used one of our panels that we have put together, specifically the lung cancer panel, to screen cell-free plasma DNA for potential resistance mutations by comparing pre-treatment and post-disease progression plasma specimens in a phase 1 clinical trial. So what they did is they took blood samples from individuals before treatment, and then they took blood samples from the same individuals after they have seen progression on this specific drug that targets T790M mutation. And so the cell-free DNA that they've isolated serves as a liquid biopsy to monitor disease progression. So how did they do this? They collected 10 to 20 cc blood from the same individuals, pre-treatment, or what they call baseline, and post-progression. They prepared plasma. They isolated cell-free DNA using the QIAAMP circulating nucleic acid kit. They enriched 20 genes that are implicated in lung cancer development and progression using the GeneRead DNA-seq lung cancer panel. They constructed libraries and they sequenced the libraries on a high-seq and achieved this ultra-deep sequencing of 30,000x. This speaks to first the power of those sequencers, but at the same time, it shows you how important targeted enrichment is, or targeted DNA sequencing, because now that you've shrunk that DNA down to those 20 genes, you can achieve this very high degree of depth. So what did they find? So when they looked at just one sample as a proof of concept, they looked at plasma samples at baseline and progression. Now they still see the T790M mutation because the population was enriched to only look at those individuals that harbor T790M mutation. However, when they looked at the progression data, so basically these are mutations that have accumulated after they saw progression, they started seeing this C797S mutation present in the progression samples, but not in the baseline samples, which was an indication to them that there's something happening, like after a certain period of time, whereby these samples are acquiring another sort of set of mutations that makes them resistant to that second-class tyrosine kinase inhibitor that has been developed to target T790M mutation. So they wanted to extend this further. Instead of going just for the proof of principle, they now wanted to look at many more samples and see if they see the same pattern or not. And they not only looked at the plasma samples, like cell-free DNA using liquid biopsies, but also they started comparing that information to what they see in the tumor itself. So they looked again at baseline and progression for the tumor itself. They looked at baseline and progression conditions using liquid biopsies. They looked at the T790M mutation. They see it across all of these different conditions, again, because it is a mutation that was enriched for. And then when they started looking at the C797S mutation, looking at two different genotypes of that same mutation, they started seeing that this mutation starts accumulating under progression conditions where it was absent under baseline conditions, indicating that this specific mutation is, at least it is associated with resistance to that second-class tyrosine kinase inhibitor that targets specifically the T790M mutation. So, as a summary and just to close off this webinar, first, targeted DNA sequencing enables precision medicine using liquid biopsies. Targeted DNA sequencing can be used to detect these low-frequency mutations. If I go back to that example, they actually started seeing this C797S mutation being present at only 1.3% of that sample. They were able to do so first because they're using a smaller panel, and now they're able to achieve this extremely high degree of depth of coverage to allow them to confidently call these low-frequency mutations from cell-free DNA. Amplicon-based enrichment using multiplex PCR delivers the high level of specificity and uniformity needed to overcome the low quality and quantity of liquid biopsy DNA. So again, you want to have an approach that gives you a very high degree of specificity and uniformity, and multiplex PCR offers you that, those requirements. Redundant and overlapping amplicon design deliver the deep coverage required for detecting low-frequency mutations. So the overlapping amplicon design also ensures that you have a continuous coverage of the regions that you're interested in, and that there are no gaps within that coverage. Liquid biopsies are an efficient way to monitor disease progression and resistance. So the example that I've shown you from Nature Medicine highlights how liquid biopsies can be used to monitor disease progression and resistance. I think that's all I have for today. Again, I want to thank you for attending, and I will turn this back to Julianne.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, What NGS platform is your target gene amplification kit compatible with?
The kits are compatible with the major sequencing platforms that exist on the market from Illumina and Ion Torrent. So you can use the kits to construct libraries to be sequenced on a HiSeq, a MiSeq, a NextSeq, or a PGM or Proton.
The next question. How different is this approach than capture-based methods for enrichment? Which approach should I use?
So there are multiple parameters on which the two different technologies differ. So, but the major difference is that the hybridization-based approaches tend to require a lot more DNA than multiplex PCR-based approaches. Multiplex PCR-based approaches require, again, anywhere between maybe 10 and 40 nanograms per sample, whereas hybridization-based approaches tend to require more, in the range of maybe 200 nanograms or so. So you need a lot more DNA, and the hybridization-based approaches tend to require more time to construct libraries and enrich the targets. Because there's a hybridization step involved, this could take anywhere between 16 hours to 24 hours in order for the hybridization to finish. So the multiplex PCR-based approach is much quicker and it requires less DNA. Which approach would you take? I would say it depends on the amount of DNA that you want to enrich, or the amount of regions that you want to enrich. If you're looking at an exome type of an approach, definitely the hybridization-based approach makes more sense. Because the multiplex PCR-based approach cannot accommodate a whole exome. But if you're looking at maybe 100 to 200 genes, I would definitely advise you to use the multiplex PCR-based approach because of the specificity and uniformity.
Will this technology be used in the future for pre-systematic detection as a screening test?
Yeah, I mean, we're always looking for sort of new applications. The main applications that we developed initially were for cancer applications, like the detection of somatic and germline mutations. But we also have panels for inherited diseases and cancer predisposition. So again, a different set of applications instead of just looking at somatic mutations in cancer. So in the future, please look for additional content. We're always developing new content for the same technology.
How much DNA can I get from a liquid biopsy?
That is going to depend on the type of sample that you're looking at. So if we look at just what's being described in the literature, you can end up anywhere between maybe 10 nanograms of DNA if you're looking at a healthy individual, up to maybe 50 or 60 nanograms of DNA, or even more if you're looking at an individual with an advanced disease. So if you're looking at, let's say, a sample that's been taken from an individual with stage 4 cancer, you're definitely going to end up with more DNA in circulation, simply because tumors are always going to shed DNA into circulation. So again, the answer is that it's going to depend, but you will definitely end up with enough DNA to do a targeted enrichment approach using multiplex PCR.
What is the minimum read depth you would suggest when dealing with liquid biopsy fDNA with targeted DNA?
I would definitely recommend at least 1500x, so 1500x for looking at somatic mutations. And again, with a targeted DNA sequencing approach, you can easily get to 1500x if you're looking at these types of mutations. If you're looking at germline mutations within cell-free DNA, which is not a very popular approach, but even if you do need to, you can bring this down to 200x. But definitely, if you're looking at somatic mutations, bring this up to at least 1500x.
What types of variants can you detect with this enrichment approach? Is this for human DNA only?
It is for human DNA only for now. We've gone for the initial applications. It's used for DNA from humans only. And the types of variants that you can detect, there are 3 different types of variants that you can look at. Single nucleotide variants like mutations or SNPs. You can look at copy number variations. So yes, you can call copy number variants from a sample using these panels. The only requirement is that you have a control sample against which all your case samples will be compared. And you can also look at small insertions and deletions, anywhere maybe between 40 and 50 bases, but nothing larger than that. You cannot call any structural variants or fusions for using these types of panels, but you can definitely look at mutations, copy number variants, and small insertions and deletions.
Can I build a panel for hotspots only?
Absolutely. So using the custom panel builder online, you can build a panel for any type of genomic regions. This could be hotspots. This could be, targeting specific exons within genes, targeting specific intronic regions or promoter regions. You can build a panel for all the coding regions within a gene or within a subset of genes, or you can target the entire gene with exons and introns. This can all be done on the custom panel builder that is present online. If you have any questions in order to use that custom panel builder, there's a link to get in touch with us. Please do so, and we'll answer any questions that you may have for you to build the panel of your choice.
What depth of coverage do you recommend?
So the depth of coverage, again, that goes back to the initial question or one of the earlier questions. If you're looking at germline mutations, definitely get to at least 200x to be confident in calling these types of variants. If you're looking at somatic mutations, definitely get at least 1500x across those regions that you're interested in order to be confident in calling somatic mutations.
Can you comment on comparisons of your panels with Illumina's TruSight 15 or 26 or cancer panels?
So I can, they differ on, again, different aspects. You have the content, and then you have the technology that's used. In terms of content, there's no sort of one content that will satisfy everyone's needs. That's why we offer the custom panel builder. I know the TruSight 15, I think, enriches 15 genes associated with cancer. If we look at our clinically relevant panel, that is a panel that enriches about maybe 15 genes or maybe 18 or 20, I can't remember the exact number, but it follows the same principle where you have a panel that enriches for genes that are implicated in different types of cancers. Then if I compare it to our actionable mutations panel, that is a hotspot panel that allows you to enrich specific regions within, let's say, the EGFR gene or KRAS gene. So the content differs, and again, the content, it might not be applicable to everyone, but you have the custom panel builder to build the panel of your choice. But then the technology also differs. We use, again, multiplex PCR-based approaches. The TruSight and TruSeq panels from Illumina use hybridization coupled with other different steps. And when we look at the differences, they will differ in terms of turnaround time and the amount of DNA that is used.
There are no more questions. I would like to once again thank Dr. Samara for his presentation. Do you have any final comments?
Yeah, I mean, it's as you can see from the questions, there are different types of approaches for targeted DNA sequencing. Again, my suggestion and my recommendation would be if you have a smaller target region that's again between maybe 100 or up to 200 genes, the multiplex PCR-based approach offers you the highest specificity and uniformity based on some papers that were published. If you want to go any larger than 200 genes or so, you might want to use some other technology. But in order to take advantage of the specificity and uniformity of multiplex PCR, we developed these products. And when it comes to the sequencing levels, do you do whole genome, whole exome, or targeted DNA sequencing? Again, remember that there is no one right or wrong approach. The right approach is going to depend on the questions that you're asking. If you're looking for every single base, Sure, you can use whole genome sequencing, but remember that there are drawbacks associated with these types of approaches whereby it's expensive and you might not have enough depth of coverage to call these low-frequency variants from such as those that you get from liquid biopsies. And that's where the targeted DNA sequencing approach can come in, which allows you to increase the depth of coverage so you become more confident in calling these types of mutations.
Thank you. I would also like to thank our sponsor, QIAGEN, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through 6 months from the lab date. You will receive an email from Labbers alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.